Okay, great. So thank you very much. Um, to make it possible online presentation for me too to present the results of our work package focus on the uh, microbiology studies, especially in its impact on the storage of hydrogen in underground storages. Do you see my presentation? Should I share my presentation?
Not yet.
So, yeah, do you see my presentation? Yeah. Okay, so the thing is, why do we concern about microorganisms when storing hydrogen in the porous underground media? So, as many studies has pointed out that despite extreme condition in underground storage, many different microorganisms can survive in subsurface environment, and in particularly with the limited electron donor, hydrogen is the key energy source for many microbial processes, including hydrogen consumption. Therefore, when we injected hydrogen into the storage facilities, we could enhance the the development of microorganisms. Especially, there is different groups of microorganisms which can consume hydrogen. For example, via the process of sulfate reduction, methanogenesis, or acetogenesis. You see in the their reaction on the slide. And as long as there is available of hydrogen, the carbonate and the sulfate, then the microbial processes can be stimulated, and this could pose many different risks for our storage. For example, the sulfate reduction process will generate the hydrogen loss and the contamination of the gas, especially contaminated with hydrogen sulfide, and many other aspects such as the corrosion and so on. And therefore, it is very important to consider this aspects of the microorganisms. And for hydrogen storage, compared to another technology, the storage of hydrogen in the geological formation, such as the rigid hydrocarbon field, are important and could also balance the demands of energy supply. However, our knowledge and the practical experience on hydrogen storage on the porous structures still remain limited. Therefore, it is very important to understand the microbial risks to ensure the success of the implementation of this technology. And in the high storage projects, I introduced by Carrie before, and the work package which focus on the microbiology studies will focus on the assessment of the potential microbial risks for hydrogen storage using the formation water from the different European porous storage. In our work package, we will focus on the characterization of microorganisms in the formation water sample. And after that, we will stimulate the activity of microorganisms under the condition which close to the storage site. And finally, the assessment of microbial risks, as well as to the development strategies to reduce the risks occur. So, so how we do that? So for the microbial characterization, we use the formation water sample, and thanks to the advisory members of different storage sites, so they have sent us different sample at least 11 storages facility with very specific condition. For example, the pressure could go up to 200 bar, and the wide range of salinity also with temperatures. So due to the diverse source of formation water sample available, so we could cover most of the condition what is essential for stimulate microbial growth and activity under the underground gas storage. So what will we do? So at the laboratory, we will perform different analytical analysis in order to assess and classify the potential biorisks for the underground gas storage. Thus include the initial examination, for example, checking the pH or visualize microorganism under microscopy and checking different chemical composition. And then we also perform the cultivation of the microorganisms, specifically focus on the hydrogen-consuming microorganisms, and also other relevant microorganisms could be involved in the microbial processes. In addition to that, the molecular analysis will also perform in the lab in order to detect the total microorganism which potentially present and also targeted on the hydrogen consuming microorganisms. So here show you the results of our molecular analysis in the 11 formation water samples, and the total microorganisms show to be present in the different formation water as archaea and or bacteria, which is quantified by the molecular analysis. And you see that over 11 samples have investigated. So we have 10 samples which is detected microorganisms by the molecular analysis. So only the sample number 5, in which the detection was below the limit of the method. And besides the total microorganisms, the molecular analysis also focus on the hydrogen-consuming microorganism, including sulfate-reducing archaea, sulfate-reducing bacteria, methanogens, and acetogens. And you could see that the main groups of hydrogen-consuming microorganisms present across different samples and with different proportion. Overall, we have at least 7 per 11 storages have contained 4 main hydrogen-consuming microbial groups, and there is 3 storages contain the low number, but at least 1 hydrogen-consuming microbial group was detected. So from the result of molecular analysis, we could first have an impression that the potential risks due to the microorganisms that are capable of using hydrogen was present in our investigated formation water sample. And besides the molecular studies, we also make an effort to enrich the viable microorganism in the formation waters, because it is possible that the molecular analysis not only detected the living cells, but also the dead cell. Therefore, this enrichment method will enable us to detect the living cells and also allow us to perform the further experiment. And especially in this experiment, the temperatures used for the incubation and the salinity was close to the original condition. And depends on the salinity level, and also the initial cell number present in the sample, the duration of the incubation will prolong. It depends on the condition, and the successful how we evaluate the successful enrichment, we will base on the activity and also the increase of the cell number. And you can see, for example, here from the original formation water, the number of cells is lower, but if we have a successful enrichment, we have an increase of the cell number, and also in this case is the enrichment of sulfate reducing bacteria, and we saw that there is the formation of iron sulfate, which is precipitate and change the color of the medium. So from this experiment, we are able to enrich microorganisms from 5 storages, and especially from these storages has been reported that hydrogen has never been injected before. So this could show that the microorganism information samples could be activated and enriched with the injection of hydrogen in the presence of the required nutrient. And this enrichment will allow us to perform the further investigation and gain more understanding on the potential microbial processes could occur under hydrogen storage underground, especially the microbial process associated with hydrogen. So the next, I will show you the stimulation of hydrogen consumption using the microorganisms which we enriched before. This following slide will also show you one case studies. But in general, we do it at high pressure test and also at the low pressure test. And for the high pressure test, so we will perform using the bioreactor unit, and for the low pressure test, we only perform in the very low volume bottle. And in both cases, we will use the enrichment that cultures what we obtained before, and the gas is 100%, and the liquid will be the formation water originally obtained. And especially in case of high pressure test, we will try to use the core materials, because we suppose that the carbon source would derive from the core materials. And for the low pressure test, besides core materials, we also use the artificial source of carbonate in order to understand more what the process could occur, because with low pressure test, there is more sampling available than the high pressure test. And especially the cultures will be incubated at the specific temperatures, but depending on the tested storage condition. And during the test, the change of the gas composition will be monitored, and the change of the pH and also the composition of hydrogen consuming group will also be monitored over the time. And this shows you the results of the hydrogen consumption test performed at the low pressure or lightly, only the overpressure of below 3 bar of the hydrogen. And you can see in the figures that there is the consumption of hydrogen due to microbial activities. And along with the hydrogen consumption, there is the formation of methane over the time. However, you could also observe that the consumption of the hydrogen reduced after 15 days of the experiment. And because in this test, we use the core materials before, so it is supposed that the carbonate which is available from the core is limited. That's why the hydrogen consumption is limited after 15 days. To confirm this assumption, we add the carbonate into the tested cultures, and we could observe that the hydrogen consumption is stimulated. Also, the formation of methane stimulated after the carbonate will make available again. And in the next figures, show you the results of the hydrogen consumption test in case we added the soluble carbonate at beginning instead of core materials. And we can see that also there is the hydrogen consumption due to microbial activities and also the methane formation. However, we also, similar to the case before, we also observed that the hydrogen consumptions stopped after 15 days. However, when we add the extra hydrogen carbonate, we didn't see any reactivated of microbial activity. So this indicated that besides carbonate, there must be another or more other factors could limit bioactivities. And I mentioned before, we also follow the pH and the cell growth over the time. And as you can see in both cases of core material and soluble carbonate addition, there is a significant increase of the pH. However, in case of the core material, we saw that the pH remained, it increased, but it remained below 9.5. And after the addition of the carbonate source, they increased up to nearly 10. And whereas when the soluble carbonate were added, we saw that the pH increased very high level, and it remained high when we have the carbonate addition, the slightly decrease of pH. So we assume that beside the carbonate limitation, the high pH could also the limit factors for the microbial activities. And we have more understanding about the process based on the hydrogen consumption test at the low pressure. At the same time, in order to come as close as possible to the real condition, we perform the hydrogen consumption test at high pressure. And as you see that during the hydrogen consumption test at the high pressure, the temperature and the pressure was monitored. In this hydrogen consumption test at high pressure, we use two different concentration of core materials to again check the assumption if the carbonate from the core has a limitation. And yes, in case of core materials, only 10%, we didn't observe any change in the gas composition. Nevertheless, we still record the active of microorganisms after the high pressure tests. So this indicates that microorganisms still active despite their exposure to high pressure of hydrogen. And so we could, with the limited of the carbon source, we could also assume that the formation of methane or the activity of methane organs still occur, but because the concentration of methane was formed, still below the detection limit, so we could not detect it. And in case when we increase the concentration of core materials up to 40%, we are able to detect methane in the gas phase. And this indicated that not only at the low pressure of hydrogen, but also at the high pressure of hydrogen, only close to storage condition, the microbial activity still occur and microorganisms actively consume hydrogen. So what we could conclude from our case studies is that, so from the whole investigation or for the 11 samples which we investigated, So we could detect microorganism in 10 per 11 samples. So this is indication of the potential risk of microorganisms. And obviously, the condition of each storage site is different. So it leads to the different composition of microorganisms. And from the hydrogen consumption test, we could see that the microbial consumption or the change of the gas composition are clearly observed. There is also some factors, including carbon availability, pH, and maybe also another different factor like temperatures and salinity may also affect the microbial activities. And for the case studies which we investigated, so we observed that the methanogenesis is the process which is active during the tested condition. So although, as you see, there is the communities of different microbial groups present, but under specific condition, specific groups of microorganisms can be stimulated. And therefore, with our observation, we would recommend that it is very important to monitor the microbial processing during the operation of underground gas storage, especially during hydrogen injection. So I would like to thank you for your attention, and also thank you very much for the high storage projects for funding us doing this such interesting investigation, and also in very interesting sample with very specific characteristic which not that easy to obtain. And yeah, again, thank you very much. If you have any questions, so feel free.